Welcome to Bio Bits and Bytes. Ever wonder what truly makes our brains tick? Maybe beyond just the buzz about neurons?
hmm
Today, we're diving deep into the world of astrocytes. You know those star-shaped cells in your brain?
Yeah, the stars of the show, finally.
Exactly. For way too long, they were sort of seen as, I don't know, bit players. But a flood of recent research shows they're absolutely central. Central to everything from how we remember things to, well, what goes wrong in brain diseases.
That's right.
Think of this as your shortcut to understanding why these unsung heroes of the brain are finally getting their moment in the spotlight. We've got a really fascinating stack of articles here, and to guide us through it all, we have an expert who can really unpack this for us.
Well, thanks for having me. It's genuinely exciting to see this field evolving so rapidly. I mean, for decades, the neuron really took center stage, right?
Don't relate.
But we're now realizing that this intricate partnership, this dance between neurons and astrocytes, is where a lot of the real action happens.
Exactly. So for years, the picture in the textbooks, it was basically astrocytes are just support staff for neurons, right? Holding things together, maybe feeding them a bit.
Pretty much, yeah. Structural support, metabolic support, that was the main story.
But our sources paint a vastly different picture, especially, in the last couple of decades.
That's right. Historically, astrocytes were viewed primarily through that lens, providing structure, fuel. But the idea that they actively participate in brain functions like, say, memory and learning, that's a relatively recent but incredibly important shift in our understanding.
And this shift, as our sources highlight, it offers some really compelling new ways to look at brain function.
Mm,
And maybe what might be going wrong in conditions that a purely neuron-focused view couldn't quite explain.
precisely. By understanding the dynamic role of astrocytes, we can gain a much more holistic view, both of a healthy brain and how neurological and psychiatric disorders might develop.
Okay, so we keep mentioning glial cells. Before we dive deeper into astrocytes, what exactly are glial cells? Where do our star-shaped friends fit in?
Right. So glial cells, or often just glia, they're the other major type of cell in our central and peripheral nervous systems, alongside the neurons.
Okay.
The name glia actually comes from the Greek word for glue, which kind of reflects that initial idea they just hold neurons together.
Their brain's glue.
Yeah, but we now know they do so much more. Crucial functions support, protection, and they actively modulate neuronal activity.
Oh.
And what's fascinating is that in many brain regions, glial cells actually outnumber neurons.
Wow, more glia than neurons. That really changes the perspective. And astrocytes are a specific kind of glial cell then.
Yes, exactly. Astrocytes are one of the most abundant and diverse types of glial cells,
hmm
specifically in the central nervous system, the brain and spinal cord.
And the star shape.
That's where they get their name, astrocyte star cell. They have this characteristic star-like shapes with all these radiating processes extending outwards. And these processes allow them to interact really intimately with, well, virtually every other cell type in the CNS. Neurons, blood vessels, other glial cells. This widespread connectivity is absolutely key to their diverse roles.
Got it. So these star-shaped cells are constantly in touch with their neighbors. Now, one of the most intriguing ways they interact with neurons is through something called gliotransmission. Can you walk us through how that works?
Certainly. So astrocytes have this huge array of receptors on their membranes, kind of like antennas, and they can detect the various signals released by neurons, neurotransmitters, for instance.
So they're listening in.
They are. And when these receptors get activated, it sets off a chain reaction inside the astrocyte. Often this leads to a rapid increase in the concentration of calcium ions within the cell.
Calcium ions, like an internal signal.
Exactly. This internal surge of calcium is what we often refer to as calcium excitability in astrocytes. Think of it as the astrocyte's way of registering, processing the messages it's getting from nearby neurons.
Calcium excitability. Okay, so it's like an internal ripple effect inside the astrocyte responding to all that neuronal chatter. And what happens once this calcium level spikes?
Well, this rise in intracellular calcium then triggers the astrocyte to release its own signaling molecules
Yeah.
back out into the space surrounding the cells.
Ah.
And this process is called gliotransmission. It's the main way astrocytes can actively communicate back to neurons and influence their activity. They're not just eavesdropping, they're really participating in the conversation.
Gliotransmission, so they're talking back. What kind of molecules are these gliotransmitters? What are they saying?
The most prominent gliotransmitters, the ones we know most about released by astrocytes, are things like glutamate, D-serine, and adenosine triphosphate, or ATP.
Okay, let's maybe quickly define each of those in this context. Glutamate, we know that's a major neurotransmitter, usually excitatory, right? What's its role when an astrocyte releases it?
Right. Glutamate is the primary excitatory neurotransmitter in the brain. It generally increases the likelihood of a neuron firing. When released by astrocytes, it can act on nearby neurons, influencing their excitability and also something called synaptic plasticity.
Synaptic plasticity. Can you define that for us?
Sure, synaptic plasticity basically refers to the ability of synapses, those connections between neurons, to strengthen or weaken over time in response to changes in neuronal activity. It's widely believed to be the fundamental mechanism underlying how we learn and form memories.
So astrocyte-released glutamate influences learning and memory circuits. Okay. What about D-serine? That one sounds less familiar.
Yeah, D-serine is an amino acid, but it acts as what we call a coagonist, specifically at NMDA receptors. These are a specific type of glutamate receptor found on neurons, and they are critically important for synaptic plasticity and learning.
So D-serine helps the main glutamate system work better for learning.
In a way, yes. By releasing D-serine, astrocytes can fine-tune the activity of these key NMDA receptors. They essentially modulate how neurons respond to glutamate, particularly in contexts important for learning and memory.
Fascinating. So astrocytes can tweak neuronal learning through both glutamate and D-serine. And the third one you mentioned was ATP. We usually think of ATP as like the cell's energy money, right?
Exactly. ATP, adenosine triphosphate, is indeed the main energy currency for cells. But it also doubles as an important signaling molecule outside the cell.
Ah, okay.
So when released by astrocytes, ATP can bind to specific receptors, called purinergic receptors, on neurons and other glial cells. And this triggers a variety of effects influencing neuronal excitability, maybe even initiating these calcium waves that spread communication between astrocytes themselves.
Oh.
Plus, ATP can be broken down outside the cell into adenosine, which has its own set of signaling roles, often involved in sort of calming neuronal activity, promoting sleepiness, things like that.
Yeah. So it's a much, much more complex signaling network than just neurons talking to neurons.
Okay, beyond this direct signaling, astrocytes also seem absolutely crucial for maintaining the brain's delicate chemical balance, especially with neurotransmitters.
Absolutely. That's a huge role. Astrocytes are vital for the homeostasis, the stable balance of neurotransmitters within the brain. They actively contribute to the removal, the breakdown or metabolism, and the reuptake of neurotransmitters from the synaptic cleft.
The synaptic cleft, could you just quickly define that?
Oh, sure. The synaptic cleft is simply that tiny, tiny gap between two neurons where communication happens. It's the space where neurotransmitters are released by one neuron and travel across to bind to receptors on the next neuron.
The gap. Got it. So astrocytes clean up that gap.
Yes, very efficiently. This is especially important for glutamate, the main excitatory one, and GABA.
GABA, that's the main inhibitory one, right? The
Yep.
brake.
Yep.
Exactly. GABA, or gamma-aminobutyric acid, is the brain's primary inhibitory neurotransmitter. It generally reduces the likelihood of a neuron firing. So astrocytes help regulate both the go signal, glutamate, and the stop signal, GABA.
How do they do that?
Well, they have specific transporter proteins on their membranes. Think of them like little vacuum cleaners. And these allow them to efficiently soak up excess glutamate and GABA from the synaptic cleft.
Okay.
This clears the signal quickly, ensuring neurons aren't overstimulated or understimulated for too long. And besides just removing them, astrocytes also metabolize them. Like, they can convert glucose into lactate, which neurons can then use as fuel. And as we'll probably get into, they're key players in recycling glutamate.
Right. Our sources really emphasize this critical role astrocytes play in preventing the toxic buildup of glutamate, something called excitotoxicity. Why is that such a big deal?
Excitotoxicity is a really big deal.
Right.
When levels of glutamate in that space outside the cells become excessively high, maybe after injury like a stroke, or in certain disease states, they can overstimulate neurons. This leads to a damaging cascade of events inside the neuron. Too much calcium influx. And that can ultimately result in neuronal dysfunction or even cell death. It's quite toxic.
And astrocytes are the main line of defense against this.
Absolutely essential. They prevent this by rapidly clearing that excess glutamate from the synaptic cleft. They are remarkably efficient. Neurons themselves do take up some glutamate, maybe about 20%, but the vast majority, around 80%, is handled by these astrocytes right there at the synapse, the perisynaptic astrocytes.
80%. Wow. Okay, and you mentioned recycling glutamate-glutamine cycle. What exactly is that, and why is it so vital?
Right, the glutamate-glutamine cycle. It's this critical metabolic partnership between neurons and astrocytes, and it's essential for sustaining glutamate neurotransmission over time.
How does it work?
So once an astrocyte takes up glutamate from the cleft, it uses a special enzyme called glutamine synthetase to convert that glutamate into glutamine.
Okay.
And what's interesting is that this enzyme, glutamine synthetase, is found almost exclusively in astrocytes, not neurons.
Uh,
Then the astrocyte releases this glutamine, and it's taken up by the neighboring neurons. Inside the neuron, the glutamine is converted back into glutamate, ready to be packaged into vesicles and used for signaling again.
so the astrocyte takes the used glutamate, converts it safely, and hands it back to the neuron in a different form to be reused.
Exactly. It ensures neurons have a constant, readily available pool of glutamate for signaling without letting the levels in the cleft get dangerously high. It's a beautiful, efficient cycle.
So astrocytes are not only the cleanup crew, but also part of the supply chain. That's quite the supportive role. Okay, let's shift gears a bit to something many of our listeners are keenly interested in, how we form memories. How do astrocytes actually contribute to this process?
Well, astrocytes contribute to memory formation through both sort of indirect mechanisms like the neurotransmitter homeostasis and energy supply we've just discussed, and also through more direct interactions right there at the synapse.
Okay, like what? Our sources mention the release of lactate in the hippocampus. That's a key brain region for memory, right?
Yes, exactly. The hippocampus is crucial. And research, mostly in animal models so far, has shown that lactate, which is a metabolic byproduct released by astrocytes, seems to play a significant role in the formation of certain types of memories, like emotional memories, and in learning generally.
Lactate, really. We usually associate that with muscle fatigue.
I know, it's surprising. But in the brain, it seems particularly important for the consolidation of long-term memories, that process where memories become stable over time. Less critical, perhaps, for short-term memory. Some studies have even shown that if you directly inject lactate into the hippocampus, you can enhance memory formation.
That is surprising. And it sort of connects to the finding that glucose, which astrocytes can convert into lactate, has also been shown to improve cognitive functions and even enhance memory in both humans and animals. It sounds like astrocytes are providing the fuel for our mental gears to turn.
Exactly. This highlights the crucial metabolic support astrocytes provide. They're supplying these essential energy substrates needed for the very energy-intensive processes of learning and memory consolidation.
What about potassium ions, K plus I's? Our sources mention astrocytes remove excess potassium from the synaptic cleft, and this contributes to something called short-term plasticity.
Yes, that's another key role. When neurons fire rapidly, like during intense activity, potassium ions build up in that tiny synaptic cleft space.
Okay.
If these potassium levels aren't tightly controlled, they can mess up normal neuronal signaling. Astrocytes express specific channels and transporters that allow them to efficiently absorb this excess potassium, helping maintain the delicate ionic balance needed for proper synaptic function. This rapid buffering of potassium contributes to short-term plasticity, those quick, temporary changes in synaptic strength that happen during learning.
There's also a study mentioned in our sources involving rats in the Morris water maze, that classic swimming test for memory.
Ah, yes, the water maze.
It showed an increase in the number of astrocytes in a specific part of the hippocampus, the CA3 region, during these learning and memory tasks. That seems like pretty direct evidence they're involved.
It does. It's an intriguing observation. It suggests that astrocytes might actually, proliferate or become more active in brain regions that are heavily engaged during learning, perhaps to provide increased metabolic and regulatory support to those hardworking neurons.
And then there's this other experiment. It sounds quite powerful, blocking gliotransmitter release, that astrocyte communication we have talked about specifically in the amygdala, which is key for emotional memory.
Right, the amygdala.
Blocking that release during memory consolidation in rats led to amnesia, and they could reverse it by injecting gliotransmitters back in.
Yes, that was a very compelling study. It focused on structures called connexin 43 hemichannels, which astrocytes use to release gliotransmitters in the basolateral amygdala.
Connexin 43 hemichannels. Can you briefly explain those?
Sure, hemichannels are like half pores or channels in the astrocyte membrane. Connexin 43 is the protein that makes them up. These hemichannels can open under certain conditions, allowing molecules like ATP and glutamate to pass from inside the astrocyte out into the extracellular space. So they're a key route for gliotransmission.
Okay, so blocking these channels stopped gliotransmission, and memory consolidation, that's the process of making memories stick, right?
Exactly. Memory consolidation is that crucial period after learning when a memory trace becomes stabilized, making it resistant to disruption and allowing for long-term storage.
So blocking gliotransmission during consolidation caused amnesia. That's huge.
It is. And the fact that the memory loss could be rescued by directly administering a cocktail of gliotransmitters, things like glutamate, D-serine, ATP, glutamine, even glycine provides really strong evidence that astrocyte communication glial transmission is necessary for this critical memory process.
Wow. Okay, one more piece of evidence. Our sources mention mice that lack a specific receptor called IP3R2. This receptor is vital for calcium release within astrocytes, and these mice showed impaired long-term memory consolidation.
Yes, the IP3R2 receptor. Can I define that quickly?
Please do.
So the IP3R2 receptor, or inositol trisphosphate receptor type 2, is a type of channel located on the endoplasmic reticulum inside the astrocyte that's an internal calcium store. When activated by signaling molecules, these receptors open and release calcium from the internal stores into the cytoplasm. This is the main way astrocytes generate those calcium signals we talked about earlier. And importantly, IP3R2 is the primary type found in astrocytes.
Okay, so mice missing this key calcium release channel had memory problems.
Exactly. This study demonstrated that while these mice could initially learn a task just fine, they had significant difficulty retaining that memory over the long term. It highlights that the dynamic calcium signaling within astrocytes isn't just for moment-to-moment chat, but it's also essential for the lasting changes in synaptic function that underlie the formation of stable long-term memories.
It's becoming so clear. Astrocytes aren't just bystanders at the synapse. They're active, crucial participants. This leads us nicely to the concept of the tripartite synapse. Can you explain what that entails?
Right, the tripartite synapse. The traditional view, for a long time focused just on the two neurons, the presynaptic neuron sending the signal and the postsynaptic neuron receiving it, a two-part conversation.
The standard picture.
Yeah. The concept of the tripartite synapse basically expands this view to recognize the absolutely critical role of the surrounding astrocyte as a third active component of this functional unit.
So it's really a three-way interaction at the most fundamental communication point in our brain. What specific roles does the astrocyte play in this tripartite setup?
Well, within this tripartite synapse, the astrocyte, with its incredibly fine processes that literally wrap around both the pre- and postsynaptic terminals, actively interacts with both neurons. As we've discussed, it acts as a key regulator, a guardian really, of the neurotransmitter environment in that synaptic cleft.
Controlling the signaling molecules.
Exactly. And these parasynaptic astrocytic processes, the bits right of the synapse, can also influence the structural stability, the dynamic changes, and even the maturation of dendritic synapses.
Dendritic synapses.
Yes. Dendritic synapses are simply the synapses, the connection points, that are formed on the dendrites of a neuron. Dendrites are those branch-like extensions of a neuron that receive signals from other neurons. So astrocytes are influencing these crucial input points.
They're right there in the thick of it, shaping how neurons connect and talk. And we know astrocytes themselves have receptors for neurotransmitters, letting them sense what's happening.
Precisely. Astrocytes possess a whole variety of membrane receptors that can detect neurotransmitters released from the presynaptic neuron. When these receptors are activated, they trigger those changes in the astrocyte's internal calcium levels, giving the astrocyte a real-time readout of synaptic activity.
And these calcium signals inside the astrocyte then trigger the selective release of gliotransmitters, letting the astrocyte talk back and influence the synapse.
Yes, those calcium signals are the primary trigger for gliotransmission. The astrocyte can then release its gliotransmitters, ATP, glutamate, D-serine, often in a very targeted way, maybe directed at specific synaptic inputs. This allows them to modulate the strength and plasticity of individual synapses, effectively fine-tuning neuronal communication.
It's incredibly sophisticated. Our sources also mention something intriguing. Some astrocytes seem to show spontaneous calcium oscillations, like internal rhythms, that might modulate neuronal activity even without direct neuronal firing.
That's a really fascinating and still very actively researched area. It suggests that astrocytes might have their own intrinsic patterns of activity, independent rhythms, that can influence the overall excitability of neuronal networks. It adds yet another layer of complexity to how our brains function, suggesting astrocytes aren't just reactive, but maybe proactive too.
It's becoming increasingly clear that astrocytes are incredibly versatile, dynamic cells. But are they all the same throughout the brain, or is there, specialization depending on the region or the type of neurons they hang out with?
Well, they are definitely not all the same. Astrocytes are far from a uniform population. They show significant diversity in their shape, their gene expression, their function depending on their location in the brain, and the specific types of neurons they associate with.
So you get specialized astrocyte types.
Exactly. This regional and cell type specificity leads to the formation of highly specialized sort of neuron glia units, where the astrocytes are really tailored to the specific needs of the neurons in their local neighborhood.
Can you give us an example of this specialization?
Certainly. One really compelling example comes from studies showing that astrocytes in the hippocampus, that key memory area, again, have this remarkable ability to actually induce neural stem cells to differentiate into new neurons.
Wow, they help make new neurons.
In the hippocampus, yes. But astrocytes taken from the spinal cord, for example, do not possess this capability. That's a fundamental difference in function based purely on their brain region of origin.
That's a striking difference. And it seems their responsiveness to neurotransmitters can also vary. They don't all listen in the same way.
That's right. The way astrocytes respond isn't fixed. It can be plastic, changeable, and specific to the type of cell signaling them. For instance, research in the neocortex, the brain's outer layer involved in higher functions, has shown that astrocytes show different patterns of calcium activity when they're stimulated by different types of inhibitory interneurons. Like, they respond differently to signals from parvalbumin-positive interneurons versus somatostatin-positive interneurons.
So they can tell different kinds of neuronal signals apart and respond accordingly.
Exactly. This differential responsiveness allows astrocytes to selectively modulate synaptic activity depending on the specific flavor of neuronal input they receive. It's very nuanced.
And it sounds like astrocytes can integrate multiple signals simultaneously, much like neurons do, deciding what to do based on the combination of inputs.
Absolutely. Just like a neuron integrates lots of synaptic inputs to decide whether to fire an action potential, astrocytes can also integrate multiple neurotransmitter signals they receive at the same time. And interestingly, this integration isn't always just a simple sum of the parts. It can be nonlinear.
Nonlinear, meaning the combined effect isn't just input A plus input B.
Right. Maybe inputs A and B together produce a much larger response than you'd expect from adding their individual effects, or maybe they cancel each other out in unexpected ways. This nonlinear signal integration allows astrocytes to perform a sophisticated level of information processing. This, in turn, influences how much and which gliotransmitters they release, leading to really fine-tuned regulation of synaptic function.
This intricate communication network involving all these diverse astrocytes is clearly essential for a healthy brain. So what happens when things go wrong with astrocytes? Particularly, let's talk about neurodegenerative diseases like Alzheimer's.
Right. Modulating the activity of astrocytes and other glial cells is increasingly being recognized as a really important potential therapeutic strategy for neurodegenerative diseases. Given their fundamental role in supporting neurons and synapses, it makes sense that when astrocytes become dysfunctional, it can significantly contribute to the progression of these devastating conditions.
Makes sense.
For instance, synaptic loss. The loss of connections between neurons is a key feature of dementia, and it actually shows a stronger correlation with cognitive decline than even the accumulation of amyloid plaques, which have traditionally been the main focus in Alzheimer's research.
So keeping synapses healthy is key, and astrocytes are crucial for that. Keeping astrocytes healthy might be a major target in fighting dementia. What are some specific ways astrocytes are implicated in Alzheimer's disease?
Well, in Alzheimer's disease brains, we observe astrocytes undergoing significant changes. They become what we call reactive astrocytes.
Reactive astrocytes. Let's define that. Is that like an astrocyte under stress?
Yes, essentially. Reactive astrocytes are astrocytes that have changed their shape, their gene expression, and their function in response to some kind of stress or injury in the brain environment, like inflammation or the presence of amyloid plaques in AD. This reaction isn't always bad, sometimes it's protective. But in chronic diseases like Alzheimer's, it often becomes detrimental.
Okay, and what happens in these reactive astrocytes in AD?
Well, they often show disruptions in their calcium signaling, what we call calcium dysregulation.
Calcium dysregulation, meaning their internal calcium signals go haywire.
Exactly. The normal patterns and levels of calcium ions get disrupted. These abnormal fluctuations or sustained high levels of calcium can really mess with their ability to support neurons properly. They also show an overactivation of certain internal signaling pathways. One really important one is the calcineurin NFAT pathway.
Calcineurin and NFA, can we define those two?
Sure. Calcineurin is an enzyme, a protein phosphatase, inside the astrocyte that gets activated when calcium levels rise. Once activated, calcineurin then activates another protein called NFA. NFA stands for nuclear factor of activated T cells, though it's found in many cell types, including astrocytes.
And what does NFA do?
NFA is a transcription factor. That means it travels into the nucleus of the astrocyte and controls which genes get turned on or off. So this calcineurin NFA pathway is a key way that calcium signals get translated into longer-term changes in astrocyte function and gene expression, and it seems to be hyperactive in AD.
So this pathway gets stuck in the on position, maybe driving harmful changes in astrocytes. And our sources say that inhibiting this pathway, specifically in astrocytes, actually helps in animal models of AD.
That's right. Studies using genetic tools or drugs to reduce the activity of calcineurin or NFAT, specifically within astrocytes in Alzheimer's mouse models, have shown benefits. Things like improved neuronal survival, better synaptic function, even reduced inflammation. It strongly suggests that these overactive astrocytes, driven by this pathway, are actively contributing to the disease pathology.
Wow. Now, there's also mention in our sources of different types of reactive astrocytes, specifically A1 and A2 phenotypes. What's the difference there?
Yeah, this is a relatively recent concept, that reactive astrocytes aren't all the same. Researchers have identified different subtypes based on their gene expression and apparent function. A1 astrocytes are generally considered to be more detrimental, particularly in neurodegenerative
The
contexts.
bad reactive astrocytes.
Often, yes. They seem to be induced primarily by inflammatory signals called cytokines, which are often released by another type of glial cell, the microglia, when they become activated.
Microglia and cytokines, quick definitions.
Sure. Microglia are the brain's resident immune cells, kind of like macrophages for the brain, constantly surveying for damage or infection.
The brain's defense force.
Exactly. And cytokines are small signaling proteins that cells use to communicate, especially during immune responses and inflammation. Think of them as messages like, danger here, or attack this. Common ones involved in inducing A1 astrocytes include things like TNF-alpha and interleukin-1 alpha.
Okay, so inflammatory signals from activated microglia push astrocytes towards this potentially harmful A1 state. What do A1 astrocytes do that's bad?
Well, A1 astrocytes seem to lose many of their normal beneficial functions. They become less effective at promoting neuronal survival, supporting synapse formation, synaptogenesis, and clearing up cellular debris through phagocytosis. And worse, they can start releasing factors that are actually toxic to neurons and also to oligodendrocytes.
Oligodendrocytes, those are the cells that make myelin, the insulation around nerve fibers.
That's right. So A1 astrocytes can contribute to damage across multiple cell types in the brain.
That sounds pretty damaging. And our sources say these A1 astrocytes are found in greater numbers in the brains of people with Alzheimer's.
Yes, studies have found that A1 astrocytes are indeed more abundant in brain regions heavily affected by Alzheimer's pathology. They're often found near amyloid plaques, and the current hypothesis is that they play a significant role in driving disease initiation and progression, partly by secreting more pro-inflammatory cytokines themselves, creating a vicious cycle.
Amyloid beta plaques are those characteristic protein clumps. What about acyl oligomers? Our sources mention these smaller forms can directly affect astrocytes too.
Right. Amyloid oligomers are smaller, soluble clumps of the amyloid beta protein. They're now considered by many researchers to be perhaps even more toxic than the large plaques.
More toxic. How do they affect astrocytes?
Well, these apoligomers can directly interact with astrocyte membranes and trigger a rapid pathological increase in intracellular calcium levels within the astrocytes.
More calcium dysregulation.
Exactly. And this calcium overload can lead to bad downstream effects, like the increased production of reactive oxygen species, or ROS.
ROS, reactive oxygen species. Those are like cellular rust, right? Damaging molecules.
Precisely. They're unstable molecules that damage proteins, lipids, DNA, contributing to oxidative stress. Furthermore, allo-oligomers messing with astrocyte calcium can also impair long-term potentiation, or LTP, at nearby synapses.
LTP, long-term potentiation. That's the cellular basis of learning and memory we talked about.
Yes, the strengthening of synaptic connections. So allo-oligomers, partly by messing with astrocytes, can directly hinder the mechanisms needed for memory formation.
So, oligomers stress astrocytes, which then contributes to memory problems. What other big picture roles do astrocytes play that get messed up in AD? Our sources mention the blood-brain barrier, BBB, and the glymphatic system.
Yes, both absolutely crucial, and astrocytes are key players in both. The blood-brain barrier, or BBB, is that protective barrier controlling what gets from the blood into the brain.
The brain security system. Define that for us.
Sure. The BBB is formed by these specialized endothelial cells lining the brain's tiny blood vessels, the capillaries. These cells have very tight connections between them, but they're tightly regulated by surrounding cells, including pericytes and, very importantly, the astrocyte end feet, specialized parts of astrocyte processes that wrap around the blood vessels. This whole unit forms a highly selective barrier, carefully controlling what passes from blood to brain.
And astrocytes are critical for maintaining that barrier.
Absolutely. They help induce and maintain the tight junctions between the endothelial cells and regulate transport across the barrier.
Okay, and the glymphatic system, that sounds newer. Is that like the brain's waste disposal system?
That's a great way to put it. The glymphatic system is a relatively recently described pathway for clearing waste products from the brain tissue. It involves the flow of cerebrospinal fluid, CSF, along channels surrounding blood vessels, moving through the brain tissue and collecting waste, including potentially toxic proteins like amyloid beta.
And how are astrocytes involved there?
They are critical. The movement of fluid in this system is heavily dependent on water channels called aquaporin 4, and these channels are highly concentrated on those astrocyte end feet that surround the blood vessels. They essentially help drive the fluid flow that clears the waste.
So if astrocytes are dysfunctional in Alzheimer's, both the brain's security system, BBB, and its waste disposal lymphatic system could be compromised.
Precisely. BBB breakdown can let harmful substances leak into the brain, causing inflammation and damage. Glymphatic impairment can lead to the buildup of toxic waste products like amyloid beta, further driving the disease. Both are increasingly recognized as important factors in AD pathogenesis, and astrocyte dysfunction likely plays a big role in both.
Astrocytes are also involved in something called neurovascular coupling, making sure active brain areas get enough blood flow. Is that affected too?
Yes, neurovascular coupling, that's the crucial process where neuronal activity triggers an increase in local blood flow to supply the needed oxygen and glucose.
How does it work, and how are astrocytes involved?
Astrocytes are thought to be key intermediaries. They sense the increased neuronal activity, perhaps through detecting glutamate or potassium release, and then they release signaling molecules that act on the smooth muscle surrounding nearby arterioles, causing them to dilate and increase blood flow.
And this process is often disrupted in Alzheimer's disease.
Yes, neurovascular coupling is frequently impaired in AD patients and animal models. This could lead to a mismatch between neuronal energy demands and blood supply, potentially starving active neurons and contributing to damage. Again, astrocyte dysfunction is likely a key contributor to this impairment.
We can even see astrocyte changes in living people now, right? P-imaging can track astrocytosis.
That's right. P-imaging, positron emission tomography, is a type of brain scan. There are now specific radioactive tracers that bind to markers upregulated in reactive astrocytes.
Astrocytosis meaning an increase in reactive astrocytes.
Yes, astrocytosis generally refers to an increase in the number or size of astrocytes, often indicating reactivity. PET scans using these tracers have shown increased astrocyte reactivity, or astrocytosis, in specific cortical regions of people who have cognitive impairment and also have a high burden of amyloid beta plaques.
So we can visualize this astrocyte reaction in the brain during the disease.
Exactly. It provides evidence in living humans that astrocytes are actively responding to the AD pathology. Some studies suggest this astroglial reactivity might even be a very early event in the disease process, maybe even preceding significant cognitive decline.
So astrocyte reactivity could be an early warning sign, perhaps initially protective, but then it might contribute to the problem, maybe even accelerate A production itself.
There's evidence suggesting that complex relationship. Early reactivity might be an attempt to contain damage, but chronic, sustained reactivity, especially that A1 type, can become maladaptive. It can lead to what's called astrocyte dystrophy, where the astrocytes themselves start to degenerate, lose their supportive functions, and ultimately contribute to neuronal damage and disease progression. It's a double-edged sword.
Given all of this, especially the calcium dysregulation aspect, targeting calcium signaling within astrocytes seems like a really plausible therapeutic strategy for Alzheimer's.
It is indeed. Because calcium signaling is so fundamental to both normal and abnormal astrocyte function, and it's clearly disrupted in Alzheimer's, finding ways to modulate it, maybe normalize it, or block specific detrimental downstream pathways like calcineurin and mFAT, could offer real therapeutic potential. It's an active area of drug development research.
It's quite sobering, really, just how deeply implicated these once, ignored star-shaped cells are in a devastating disease like Alzheimer's. And unfortunately, their involvement doesn't stop there. Our sources also touch on their role in mental disorders, specifically major depressive disorder, or MDD.
Yes, that's another important area where astrocyte research is shedding new light. Over the last decade or so, evidence has really accumulated highlighting the involvement of astrocytes in the pathophysiology of psychiatric conditions like MDD. Studies are finding significant morphological and functional alterations in astrocytes in patients with depression.
Like what kind of alterations? Our sources mention things like decreased astrocyte cell numbers and lower levels of a protein called GFP in key brain areas like the hippocampus and prefrontal cortex, based on postmortem studies.
That's correct. GFAP, or glial fibrillary acidic protein, is a major structural protein in astrocytes, often used as a marker for them. Multiple post-mortem studies of individuals who suffer from MDD have reported consistent reductions in both the density of astrocytes and the levels of GFA protein in these mood-regulating brain regions.
And these findings from human brains are mirrored in animal models, right? Animals subjected to chronic stress, which often develop depressive-like behaviors.
Yes, exactly. Many animal models that use chronic stress exposure to induce depressive-like states also show similar reductions in astrocyte numbers and GFA levels in the hippocampus and prefrontal cortex. This convergence of evidence from humans and animals really strengthens the link between astrocyte changes and depression.
Interestingly, our sources even suggest that if you directly impair astrocyte function in animal models, you can actually cause depressive-like behaviors. That sounds like pretty strong evidence for a causal role.
It does point towards that, yes. Studies using pharmacological tools or genetic manipulations that specifically target and impair certain astrocyte functions, like their energy metabolism or glutamate uptake, have demonstrated that disrupting these cells can, by itself, lead to the emergence of behaviors in animals that resemble depression, such as anhedonia or behavioral despair. It suggests astrocytes play an active role in maintaining normal mood states.
And one key mechanism seems to be, again, related to glutamate, a disruption of glutamate turnover between astrocytes and neurons in MDB.
Exactly. As we've hammered home, astrocytes are critical for regulating glutamate levels in the synapse. In MDD, there's growing evidence for an imbalance in this glutamatergic system. Altered glutamate signaling is thought to be a major contributor to the underlying mechanisms of depression, and astrocyte dysfunction appears to be a key part of that story.
And specifically, the expression of molecules within astrocytes that handle glutamate, things like EAT1, ET2, and GS, is found to be decreased in MDD. Can you remind us what those are?
Sure. EDT1 and ET2 are the excitatory amino acid transporters. They're those vacuum cleaners on the astrocyte membrane that suck up glutamate from the synaptic cleft.
The glutamate transporters.
Right. And GS is glutamine synthetase. That's the enzyme inside the astrocyte that converts the glutamate into glutamine for recycling, part of that glutamate-glutamine cycle.
So lower levels of these key proteins mean astrocytes are less efficient at clearing and recycling glutamate in depression.
That's the implication, yes. If astrocytes aren't effectively managing glutamate levels, it could lead to subtle but chronic alterations in glutamatergic signaling that contribute to the mood disturbances seen in MDD.
And this idea gets a boost from the finding that Willis-Ols, a drug known to enhance the function of glutamate transporters, possibly those on astrocytes, has shown antidepressant-like effects in animal models.
Yes, that's an interesting piece of pharmacological evidence. Riluzole, which is actually approved for treating ALS, seemed to work at least partly by boosting glutamate uptake. Its effectiveness in preclinical models of depression suggests that enhancing astrocyte-mediated glutamate clearance could indeed be a valid therapeutic strategy for treating MDD. It opens up new possibilities beyond the traditional serotonin-focused antidepressants.
It's truly remarkable how these once overlooked, star-shaped cells are now emerging as central players in such a huge range of brain functions and, unfortunately, brain disorders. Okay, let's just take a moment to really appreciate what we've uncovered in this deep dive. Astrocytes, they are so much more than just brain glue, aren't they?
Absolutely. They're active communicators. They're critical regulators of the entire brain environment. Key players in everything from how we learn and remember to the development of these devastating neurological and psychiatric disorders.
Their intricate and dynamic interactions with neurons are just fundamental to a healthy, functioning brain.
Completely fundamental.
And here's where it gets really interesting, I think. This complex dance between neurons and astrocytes, the diverse chemical signals, the gliotransmitters they use to talk, and the multitude of ways astrocytes support and even actively influence neuronal function. A lot of this is still being uncovered, right? This field is moving fast.
It's incredibly dynamic. Every year brings new discoveries about astrocyte diversity, their signaling capabilities, their roles in different brain circuits. It's a very exciting time for glial biology.
And the more we learn about this intricate partnership, the more potential we seem to unlock for understanding and hopefully treating a wide range of brain conditions.
It's the real hope, isn't it?
So what does all of this mean for you, our listener? Well, Understanding the central role of astrocytes, it really opens up exciting new perspectives on how your own brain works. And it offers these promising new avenues for developing treatments for brain diseases, maybe even for enhancing cognitive function down the line. The fact that targeting these cells, astrocytes, is actually showing promise in conditions as tough as Alzheimer's and depression, it really highlights their significant therapeutic potential.
It really does offer a sense of hope for developing novel therapeutic strategies, maybe ones that work alongside or even better than traditional approaches that have focused almost exclusively on neurons for so long.
For those of you listening who want to delve even deeper, you might consider exploring maybe the specific intracellular signaling pathways within astrocytes, like that calcineurin NFAT pathway, or look into the different types of gliotransmitters and their precise effects on different types of neurons, and definitely keep an eye on the ongoing research into astrocyte-targeted therapies.
Yeah. And investigating the fascinating diversity of astrocytes, how different subtypes and different brain regions have specialized functions, that's another huge area. How do astrocytes in the visual cortex differ from those in the hippocampus, for example? And how does that relate to function and disease? So much still to learn.
Okay, here's a final provocative thought for you to take away. Given just how crucial astrocytes are for pretty much all aspects of brain function, and the fact that they're implicated in so many different disorders, could optimizing astrocyte health and function be a key? A key to unlocking new levels of cognitive ability, maybe building resilience against stress or aging, and perhaps even preventing or mitigating a wide range of neurological and psychiatric conditions.
That is a very compelling question. The potential for harnessing the power of these star-shaped cells for repair, for resilience, for optimal function, it feels immense. Future research will undoubtedly continue to explore exactly that.
It really changes how you think about the brain. Thank you so much for joining us on BioBits and Bytes for this deep dive into the absolutely fascinating world of astrocytes.
It was my pleasure. Thanks for the great conversation.